Hello again. Now, okay, continuing with the genetic code. If you feel that this slide is a bit confusing, so just forget about it, because it's meant to tell you what scientists were thinking at that time. You know, if one is the code, then you only have even 5 amino acids. It's not enough. You know, the codes can be ambiguous. It has to be ambiguous because you only have one, right? A codes for something, so that means one must code for 5 bases at least, right? Sorry, one base must code for 5 amino acids at least, because you only have 4 bases. So, right? You see what I'm trying to say? You have 4 bases. You need 20 amino acids. If this is just an if question, if each is only used as a singlet code, single, so A codes for so that means you have 20 amino acids. That means one base must determine 5 amino acids, right? So that's why it says it cannot be. It's ambiguous. You can't really have one base that determines 5 amino acids. Which one, right? So that's what it's saying. So if you find it confusing, just ignore it. So these are the people that's involved in the cracking, the understanding the genetic code. There are various experiments. It went on for years in the 1960s until the Corona, Ochoa, and these are the Nuremberg, the very important discoveries that allowed them to also win the Nobel Prize. Now, here is the genetic code, or sorry, this is a codon table. If you look at the codon table, it is standard. It is always arranged as UCAG, UCAG, UCAG. You know, so this is standard. Any book, you don't make your own table and you know, you'll get confused. So it's always UCAG, UCAG, and UCAG. All right. So this will be the first position, second position, and third position. All right. Now we'll look at that in the code in more detail later. So you have 64 codes, right? For each position there are 4 options: A, T, G, and C. So you have 4 and 4, so that you get 64 codes, in which only 61 is used for coding amino acids. 3 codes are actually stop codons. Alright, so you have the UAA, UAG, UGA. They have actually given names for it. UAA is ochre, UAG is amber, and UGA is opal. They are stop codons. They are also sometimes called termination codons, and sometimes they are called nonsense codons. There's only one code for starting, which is the AUG. Now, that is the general idea, but as you do more and more research, you'll find that that's not the main or the only rule. This you'll find this if you study genetics further. You'll find that, you know, what you study in the first year, second year, yes, it's true most of the time, right? But there are exceptions. So yes, AUG is the start codon most of the time, and it specifies methionine. So this is the slide talks about degeneracy. Let's just look at this. Okay, now this is the same codon table that I showed you earlier. So look, it's been grouped into several shapes and sizes. Now the blue, oblong, and circle are the codons that have the amino acids that have 6 codons. Leucine, it has 6 codons. 1, 2, 3, 4, 5, 6. Serine has 6 codons. 1, 2, 3, 4, 5, 6. Can you see that? Arginine has 6 as well. 1, 2, 3, 4, 5, 6. Alright, so these are blue. So these are leucine, serine, and arginine have 6 codons each. And then if you look at this pink, what's the difference here in pink is that the fourth position, sorry, the third, sorry, codons only have 3. bases. So the third position is irrelevant, or it doesn't matter if it is A, T, C's or G's, or in this case it's either A, C, U or G. It doesn't matter because it means the same. Look, CCU, CCC, CCA, CCG is proline. ACU, ACC, ACA, ACG is threonine. Okay? So these are the pink ones. GUU, GUC, GUA, GUG are all coding for valine, right? So it's the same one here. So you've got one that's got 6 amino acids, You've got an amino acids that have 6 codons. You have amino acids that have 4 codons. All right. Now, what's not marked here: histidine, glutamine, asparagine, lysine, aspartic acid. Sorry, that's glycine. That's glutamine. That's glutamic acid. Now, if you look at this, what's the difference? Is that the third position again, it's different. It gives a different meaning if it's either purine or if it's a pyrimidine. Look, if it's Ca pyrimidine, it's histidine. If it's Ca purine, it's glutamine. If it's Au pyrimidine, it's asparagine. If it is Aa pyrimidine is asparagine. If it is Aa purine, it's lysine. Can you see that? Ga pyrimidine, AAS asparagine. Aspartic acid, and then Ga purine is glutamic acid. See the difference? The same up here as well. Ug pyrimidine is cysteine. Ua pyrimidine is tyrosine. All right, so you can see Uu pyrimidine is phenylalanine. I get confused as well. All right, so you have amino acids that have 6 codons, you have amino acids that have 4 codons, and you have codons that differ. That gives a different meaning if they are either purine or pyrimidine at the third position. And then of course you have the stop codons. Methionine and tryptophan each have only 1 codon. Okay, that's it. That's about the codon table. Okay, I'll stop now and I'll continue to the next slide.